Alrighty. Good afternoon, everyone. Welcome to today's Pain Research Forum webinar. I'm Paramita Basu. I'm a member of Pain Research Forum's Early Career Contributor. So today our webinar would be on pain and comorbid affective disorders with three outstanding speakers and Dr. Laura Maile from Washington University. She will be moderating this webinar. So before we start today's event, I'm going to go over the two upcoming events. The first one is our PRF Journal Club that is scheduled next week, May 28th. So Dr. Ruth Quintana from University of Florida, she's going to present this paper. So we have this QR code as well as this is posted on our PRF website. So please go ahead and register for this event. The next event is our next month's webinar. That's going to be on orofacial pain. Again, use this QR code as well as it's on our main PRF website to register. So just remember, for all the events, you have to register in order to get the link to attend that particular event, whether it's a webinar or journal club. A few house rules for today's webinar. You can use the chat box or the Q&A box to type in your questions, and the moderator will deliver those questions to our speakers. The view option menu at the top of the window will let you zoom in on the shared screen. And today's webinar will be recorded, and those recordings will be available on the Pain Research Forum website as well as our PRF YouTube channel. So without any further delay, I will turn it over to Laura.
Thank you, Paramita. Thanks again, everyone, for joining us today for this webinar hosted by Pain Research Forum. I'll be the host for this installment of the PRF webinar series. So my name is Laura Maile, and I'm a postdoctoral research scholar at Washington University in St. Louis, where I work in the lab of Dr. Reem Alhassani. I finished my PhD last year at the University of Cincinnati under the mentorship of Dr. Steve Davidson, where I studied pain and chronic stress and the neural circuitry that underlies these often comorbid conditions. So due to that, I'm really excited about our excellent lineup of speakers today who will be discussing pain and affective disorder comorbidities. So they include Dr. Andrea Nackley from Duke University, Dr. Ipek Yalcin from Strasbourg in France, and Dr. David Finn from University of Galway in Ireland. So I'll introduce each speaker one by one, and we'll have time for questions following each speaker. So please feel free to enter your questions into the Q&A at any point, and then I'll deliver those following each speaker, and hopefully time for some additional discussion at the end. So without further ado, our first panelist today is Dr. Andrea Nackley. Dr. Nackley is an associate professor and associate director of the Center for Translational Pain Medicine in the Department of Anesthesiology at Duke University. She's the director of Translational Pain Research Laboratory, where her program marries pain neurobiology, behavioral pharmacology, and molecular genetics in mouse and man to better understand mechanisms of and identify treatments for chronic primary overlapping pain conditions. Her lab was the first to demonstrate a critical role for the peripheral adrenergic receptor beta 3 in the development of chronic pain and neural inflammation, which remains a primary research focus. In recognition of her scholarly activity in the pain field, she received the John C. Liebeskind Career Scholar Award from the American Pain Society. Dr. Nackley served on the board of directors for USASP and was recently recognized as a USASP Fellow, which is the highest class of membership within the association and acknowledgement of outstanding and continuing work in the pain field. So without further ado, I'll turn it over to Dr. Nackley. Thank you so much.
Thank you so much for the kind introduction and for the invitation to speak today and share my lab's research. And I think now, great. So my talk is going to be focused on chronic primary overlapping pain conditions, or CPPCs, and comorbid mood and their mechanisms and models. So the term chronic primary pain conditions, or CPPCs, refers to these 10 conditions shown here, including temporomandibular disorder, tension type headache, and migraine that affect the face, head, and neck sites, endometriosis, vulvodynia, interstitial cystitis, and irritable bowel syndrome that affect abdominal sites, low back pain, and chronic fatigue syndrome and fibromyalgia that affect the whole body. These conditions are characterized by pain lasting 3 months or longer in the absence of obvious tissue damage. And this is in contrast to neuropathic or inflammatory pains where the pain occurs secondary to injury or antigen exposure. These conditions are quite prevalent, affecting 1 in 3 individuals in the US and 1 in 5 individuals worldwide, and they predominantly affect females. And the overlap is really the norm here, ranging from 20 to 90%, depending on the index condition that is being measured and the method of determining overlap. And this really suggests a common etiology among these conditions that affect different body sites. And, in line with today's theme, we know that these conditions are linked to mood disorders. And this figure here was taken from a recent article from Lee and Houghton's labs, where they were looking at the pain trajectories and the relationship with mood disorders in 2 large cohorts. The All of Us cohort, which included 340,000 individuals, and the Mayo Data Warehouse, which included data from 4 million individuals. And as you can see, these chronic primary pain conditions are all linked to, or most of them are linked to anxiety disorder and depressive disorder. So in fact, over 75% of these conditions have at least one mood disorder. So CPPCs are linked to enhanced catecholamine tone. So while their origins don't have an organic pathogenesis, they are linked to these genetic and environmental factors that enhance catecholamine tone. Including variants in the gene encoding catecholmethyltransferase, or COMT, that lead to low activity of the enzyme that clears catecholamines, and this results in increased levels of circulating norepinephrine and epinephrine. And then the effects of CMT genotype are modified by environmental factors such as injury and stress that can promote the release of norepinephrine from sympathetic terminals. So the effect of CMT genotype and stress on CPPC risk is shown here in results from a case control study where we enrolled 550 individuals with at least one CPPC and 250 pain-free controls. So perceived stress is shown on the x-axis and the probability of developing a CPPC on the y-axis. And as you can see, those that report high stress are more likely to develop a CPPC if they have 1 or 2 copies of the RS4680 allele, this minor variant that codes for reduced activity of the CMT enzyme. So we also know that these conditions are associated with low-grade inflammation. In fact, there's over 1,000 publications that have demonstrated increased levels of pro-inflammatory cytokines and chemokines in patients with CPPCs, as well as mood disorders. So we are interested in looking at the relationship between CMT genotype and stress on these inflammatory mediators in our CPPC cohort. And this work was done by Margarita Klein, who's an analyst in the lab. So of the 40 inflammatory mediators that we screened in plasma samples from individuals with CPPCs or pain-free controls, we identified 2 that were linked to CMT genotype and stress. So interleukin-6 and interleukin-17A. So these are both pro-inflammatory cytokines that can promote nociception by directly binding to receptors on nociceptor terminals, or they can also regulate the transcription of other inflammatory mediators. And in fact, IL-17A has been shown to positively regulate IL-6. So as you can see, individuals that report low stress, shown in the white bars, don't have, and there's no effect of genotype on their IL-6 or IL-17 levels, while those in the high stress group, among these individuals, you can see a dose-dependent increase in IL-6 and IL-17A, depending on the number of copies of the minor allele. And this is true for individuals with CPPCs, but not for pain-free controls. So building on these clinical data, we developed a mouse model of CPPCs for understanding mechanisms and screening treatments. So here we take COMT heterozygous mice that carry a genetic predisposition towards pain and expose them to a 3-day swim stress paradigm followed by a molar extraction. And then behavior is assessed prior to and over the course of 1 month. And then we also evaluated norepinephrine and cytokine biomarkers. So first, looking at the effect of COMT genotype and stress on local pain, we found well, actually, the first 2 panels are just showing the effect of the molar extraction alone in wild-type mice. So you can see that the molar extraction alone produces a very transient or facial hyperalgesia that resolves after day 3. But then if we look at individuals, mice undergoing the stress as well, we can see that compared to those in the sham groups, wild-type mice undergoing stress in addition to the molar extraction have more pronounced pain of greater magnitude lasting 14 days. So these data here show the effect of stress in and of itself on post-surgical pain. Then the CMT het mice undergoing the intervention showed the most dramatic increase in orofacial hyperalgesia, and the female's pain persists for at least 28 days. We also found that the female but not male mice exhibited orofacial grimace, which is an indicator of spontaneous pain. Then we evaluated the effect of CMT genotype and stress on pain at remote body sites. And we found that, again, compared to the wild-type and CMT het sham controls, the wild-type mice undergoing the intervention show increase in hyperalgesia at plantar, abdominal, and back sites. And, again, the CMT het mice show the greatest magnitude of pain at all 3 body sites. And these data are collected from females. And then males show a similar pain trajectory, although the magnitude of pain at abdominal and back sites was not as great as in females. Recent data collected from Savannah Dewberry, who's a postdoctoral fellow in the lab, demonstrated that mice exhibit visceral pain in addition to widespread musculoskeletal pain. So specifically, they have decreased visceromodal response thresholds and increased grimace following a colon distension. And they have increased gut motility at later time points, and this is in line with an IBS-D phenotype. So looking at biomarkers, similar to patients, we see that the CPPC mice have an increase in circulating levels of norepinephrine as early as day 7 that persist at day 28. And they also have an increased circulating level of IL-6 and IL-17A. Remarkably, of the 35 inflammatory mediators that we measured in this cohort, the same 2 that popped up in the clinical our clinical cohort were these, and so that was quite remarkable. And the increase in IL-6 was driven largely by females. So we wanted to extend the time course of pain and also measure depression and anxiety in these mice. And so to be in line with ISP's definition, we should measure 3 months, but we ended up measuring out for 12 months in these mice. And so we can see that when we look at the extended time course for pain, that females exhibit quite robust and duration of pain lasting at least 48 weeks, so about 1 year. And in comparison, the males resolved by 32 weeks, so around 7 months. When we looked at depressive behavior, we did not see any differences in depressive behavior, any indication of depressive behavior until 8 weeks. So this is 2 months after the pain. And we can see in the females that the depressive behavior following its onset persists for the full 48 weeks. While in the males, it resolves by week 32, which is in line with the trajectory of the pain behavior. We also found that female, but not male mice in the CPC group exhibit anxiety at 44 weeks, so towards the end of this time course. So using this mouse model and following a couple of decades of pharmacologic and other studies, we identified adrenergic receptor beta 3 or ADRB3 as a primary receptor that is mediating chronic primary pain conditions. So this is a G protein coupled receptor that can bind to GS and GI proteins to lead to phosphorylation of intercellular kinases and MAP kinases, and can promote the transcription of pro-inflammatory mediators. So ADRB3 is predominantly expressed on adipocytes or fat cells in peripheral tissues. And adipocytes compose about 2/3 of the cells in adipose tissue, with the other third including stem cells, immune cells, and endothelial cells. We know that adipose tissue is well known for regulating appetite and energy and temperature, insulin and glucose homeostasis, and lipid metabolism, but what could it have to do with pain? So structurally speaking, we know that adipose tissue receives input from sympathetic nerves that originate from pain and stress relevant nuclei in the brain. So this live mouse multiphoton microscopy image here shows TH positive catecholaminergic fibers innervating the inguinal fat pad. We also know that adipose is rich with sensory innervation. And so this confocal image here shows adipocytes engulfed by PGP 9.5 positive neurites. Functionally speaking, adipose tissue is a key contributor to inflammation through secretion of adipokines and cytokines, predominantly IL-6. In fact, adipose tissue has been shown to produce 30% of the IL-6 in circulation. Adipose can also promote neuroinflammation through humoral means, and this can lead to crossing of the inflammatory mediators across the blood-brain barrier, so that it can regulate the activity of neurons and glial cells. So to directly test the role of ADRB3 on adipocytes in pain and related mood disorders, we generated these conditional adipocytes ADRB3 knockout mice. So ADRB3 is knocked out specifically of adipocytes, and these mice were crossed with COMT het mice or littermate controls. And we validated this mouse using a number of approaches, including in situ, RNA-scope. And as you can see here, ADRB3 is absent in adiponectin positive adipocytes in the conditional knockout mice. So using these mice, we found that compared to those with intact ADRB3 expression, those deficient in ADRB3, specifically in adipocytes, were protected from developing pain at plantar as well as abdominal and back sites. And then when we looked at depressive behavior, we see that these mice are also protected against development of depressive behavior over the course of the 84 days that we measured, so over around three months. Interestingly, they seem to have, they weren't significant differences at baseline, but even the baseline levels of these mice was a little bit lower. So maybe even in the absence of stress, they're less prone to depressive behavior. In summary, COMT genotype and environmental stress factors can lead to increased circulating levels of catecholamines that can promote multi-site body pain and comorbid depression and behavior, likely through inflammation in part. We've developed a clinically relevant mouse model of these conditions where mice exhibit multi-site pain lasting over 3 months, comorbid depressive behavior beginning after pain onset, and increased levels of norepinephrine and cytokine biomarkers. In line with patients, these mice exhibit sexual dimorphism in symptom duration and immune profiles, so that the pain and depressive behavior lasts 7 months in males, but persists for 12 months in females. Also, female but not male mice exhibit anxiety at 12 months and have increased levels of IL-6. So and finally, multi-site body pain and depressive behavior is mediated by adipocyte ADRB3. And these work are online or ongoing line of research for our lab. So I'll stop there, and I thank most of, or acknowledge most of my lab members along the way, and then I'd be happy to take any questions we have time for now.
Great. Thank you so much for that, Dr. Nackley. We do have a couple questions. I'll just start with the first one. Is it possible that the effects of stress on widespread pain reflect that the mice are hypervigilant? Have you tested the response to non-noxious or non-tactile stimuli like sound or startle response after stress?
So after the stress, so we have not looked at startle responses. We haven't really looked at hypervigilance by looking at other types of like sensory stimuli. I mean, aside from like mechanical stimuli, they don't seem to be hyper responsive to thermal stimuli. So it does seem to be more mechanical, which is in line with the clinical phenotype of these patients. But I think that is an important point and something that would be interesting to look at, maybe looking at sound and light sensitivity. So that's a really good point. Good question.
And we'll do one more for now. This is from Dr. Adam Dorson at WashU. Is ADRB3 differentially expressed in adipose tissue between males and females? What about in human adipose tissue between males and females?
So another really great question. It is differentially expressed. So ADRB3 is actually expressed a little bit more highly in males versus females. And that's something that we definitely need to take into account. We're in the process of developing ADRB3 antagonists for pain. So yeah, this is something that we're also exploring, but yes. Good question.
Great. Okay, just for the sake of time, we're going to move on to our second speaker now, and then we'll get to additional questions at the end. And I believe our speakers also have access to the Q&A if you would prefer to type answers to questions that we don't get to. So up next, we have Dr. Ipek Yalcin, who is a research director at CNRS in France. She leads the pain and psychopathology team at the Institute of Cellular and Integrative Neuroscience in Strasbourg. She's also an adjunct professor in the Department of Psychiatry and Neuroscience, Université Laval. She obtained a tenure research position at CNRS in 2010, where she works on the comorbidity between chronic pain and mood disorders. She's authored 72 publications, 6 book chapters, and holds 2 patents. She's received several awards, including the Young Investigator Award from NRSAD, Young Talent Award from the University of Strasbourg, and several others. She's also a senior editor of neuropharmacology and is on the editorial boards of additional journals in the pain field. So I'll turn it over to you, and thank you so much, Dr. Yalcin.
Thank you for the invitation. I will try to share my screen. Thank you for inviting me to this webinar, which perfectly aligns with my research interest. Indeed, my lab last 15 years, we are interested in better understanding the vicious cycles between chronic pain and mood disorders. So hopefully Andrea did a great job for the introduction, so I can skip rapidly this part. So what I would like to talk about is that you all know that when pain becomes chronic, it's frequently comorbid with sleep disturbance, cognitive deficits, impaired word processing, but also mood disorders like anxiety and depression. As shown by Andrea, this is almost 50% of chronic pain patients develop mood disorders during their lifespan. And this is around 37% for neuropathic pain patients, but it can reach to 80% in fibromyalgia patients. So it means that chronic pain patients have almost 3 times more risk to develop mood disorders than the normal population. On the other hand, stress, especially chronic stress, can also increase the risk of the development of chronic pain, and it can also exacerbate and prolong the chronic pain symptoms. It's once again around 50% of patients with mood disorders like anxiety disorders or major depressive disorder may develop chronic pain symptoms during their lifespan. So our hypothesis in the lab was there might be some shared neuroanatomical substrate or molecular mechanisms between chronic pain and mood disorders. So we developed our research around 3 axes to see whether if we can break this vicious cycle between chronic pain and mood disorder. So the first step was just to characterize the animal model that we have in the lab to see whether we can really model the anxiety and depression in neuropathic pain condition. Second step was just after characterizing our model, if we can dig the underlying mechanism of this comorbidity. And the ultimate goal is the development of novel treatment strategies. And all along these experiments, we really try to take into consideration sex differences that I'm going to present you in next slides. So from our side, I mean, in our lab, we will try to put a lot of efforts to characterize a neuropathic pain model. But while we were doing, we were focusing on the neuropathic pain, the whole field focusing on the comorbidity between chronic pain and mood disorder also try to develop new models. So I take these images from some reviews where they very nicely demonstrate that different animal models like the nerve injuries or the inflammatory pain models or chemotherapeutic induced pain models or transgenic animals can be used to model the comorbidity between chronic pain and mood disorders. And also generally to test anxiety or depressive-like behavior, we were previously using the classical behavioral tests like the elevated plasmase, dark light, cross preference, or splash test. But last 10 years, we start to develop more and more the tools to assess the behavioral of the animals in their own environment, like the live mouse tracking, IntelliCages, or PhenoWorld. So these tests help us to really assess the behavior of these animals in their own environment, which is, I think, added value. So on the other hand, we are also interested in the impact of stress on pain chronification in animals. So several tests like the acute stress, like the restraint stress or repeated restraint stress, chronic mild stress, social defeat, or early life stress are used to study the impact of the stress on mechanical and thermal hypersensitivity or sensitivity, and also the pain chronification. So in my lab, as I mentioned, we are mainly focusing on neuropathic pain, and we use a cuff model, which consists of implanting a polyethylene tube around the main branch of sciatic nerve. So this procedure, as you see in the male animals, induce long-lasting mechanical hypersensitivity, which lasts around 14 weeks, and a very brief thermal hypersensitivity. And interestingly, the animals start to show anxiety-like behavior 2 or 3 weeks after the surgery, and then they start to develop depressive-like behavior, which persists up to 4 months. And recently, we decided to also characterize this model in female, and we realized that the mechanical hypersensitivity is longer or prolonged, as well as the thermal hypersensitivity prolonged in female animals compared to male animals. And interestingly, we didn't really observe any anxiety-like behavior in female, but we do see a depressive-like behavior, which is very similar what we observed in male animals. But what was interesting is with these all experiments that the anxiety and depressive-like behavior developed in a time-dependent manner in a neuropathic pain condition. So once we characterize these model in both male and female, our next question was, what are the underlying mechanisms? And for this part, the first other question was, which brain area is involved in this comorbidity? So among the brain areas, we are very much interested in the anterior cingulate cortex, as it's involved in both pain processing and emotional processing. And some years ago, we very basically lesion this brain area, and we observed that the lesion of the anterior cingulate cortex can block the anxiety and depressive-like behavior without affecting the mechanical hypersensitivity. So we then wonder whether what's happening in the anterior cingulate cortex. So we did some extracellular in vivo electrophysiological recording, and we realized that the animals, the ACC of the animals are hyperactive only when we observed anxiety and depressive-like behavior at the same time. The next question was that if this hyperactivity is the reason of the anxiety and depressive-like behavior that we observed in our neuropathic pain model, so what we did is that using optogenetic approach, we inhibit the excitation or excitability of the ACC neurons, and we observed that this is enough to block anxiety and depressive-like behavior. So it seems that there's a causal link between the ACC hyperactivity and the behavioral output. And we also did some molecular analysis within the anterior cingulate cortex at different time points after the neuropathic pain induction. And what we realized that we have several gene expression alterations in the anterior cingulate cortex when we observed comorbidity between chronic pain and mood disorders. So all these data really reinforce our hypothesis that the ACC is a critical hub for the comorbidity between chronic pain and mood disorder. So while I was obsessed with the anterior cingulate cortex, my colleagues were working on other brain areas. So there are several groups show that the involvement of other brain areas, other cortical areas like the MPFC insular cortex, subcortical areas like amygdala, habenula, and the brainstem structures like the locus coeruleus and ventral tegmental area. So altogether, these results show that there are several changes in the brain occurs when the animals start to develop depressive-like behavior. For instance, there's a huge alterations in the noradrenergic system, and there's a increased bursting activity that is observed by one of my collaborator, Esther Berocosa, and they also identified that there's an increased transporters in the locus coeruleus. And then other groups show that there is a where I arrived to Andrea's topic is that where they demonstrated that there's an imbalance between pro-inflammatory cytokines and anti-inflammatory cytokines in different brain areas, including the amygdala. And interestingly, there are several studies link the epigenetic alterations with the comorbidity challenge. So what I want to emphasize here is that altogether in the field last 10 years, we really go further and we start to really identify different brain areas and different mechanisms involved in this comorbidity. But we all know that a single brain structure per se cannot explain this comorbidity challenge, right? So the neuroscience go to better understand the circuitry of the pathologies, and our field also move and follow this tendency, I can say, and we also start interested to decipher the circuitries involved in this comorbidity. So my lab also followed this tendency last 5 years, and we decided to dissect the ACC circuits that might involve in this comorbidity. So we first focus on the connections between the basolateral amygdala and the anterior cingulate pathway. Indeed, we all know that there's a very nice projections, robust projections from the BLA to the anterior cingulate cortex. And we also did in my lab, in collaboration with another institute, resting state fMRI at different time points after neuropathic pain induction. And we observed that there's a strong functional connectivity between amygdala and the anterior cingulate cortex only when we observed comorbidity between chronic pain and mood disorder. So it seems that this pathway, BLA-ACC pathway, is hyperactive. So what we did is that we inject a virus archaeorhodopsin, we inject archaeorhodopsin into the BLA, and we stimulate the terminals in the anterior cingulate cortex, and we block the BLA-ACC pathway. And what we observed is that the animals do not show any more depressive-like behavior, even though they have a mechanical hypersensitivity. So it seems that this BLA-ACC pathway is involved specifically in chronic pain-induced depression. We then wonder where these information can be conveyed. So BLA bring this information to ACC, but which brain area that ACC can send this information? So we return back to our fMRI data, and we've identified that the mesolimbic pathway is very much hyperactive. The connectivity between the mesolimbic areas and the anterior cingulate cortex is very much active. So we wonder whether if we may directly focus on the ACC neurons that are projecting to habenula, is it possible to affect the behavioral outcome? So this is what we did. We inhibit the ACC neurons projecting to habenula, and we observe that this is sufficient to block the depressive-like behavior. So while I was obsessed again with the ACC and its circuitry, so again, our colleagues were working on the other brain structure. So I want to put three different review. So one comes from the David's lab, and one coming from my lab, and another one from the another group from China, Guo, and collaborators. You can clearly see that we really start to find the puzzles, and we start to connect these puzzles, and we start to really understand the circuitries involved in this comorbidity. But you can clearly see that it's not that easy, and it's even when I look, it's overwhelming. But the good news is that we really all together start to decipher these circuitries, which might help us to go one step further with but all together. So if I wrap up, what I would like to emphasize today is that, one hand, it is possible to study the anxiety and depressive-like consequences in animal model by taking into consideration time factor. This is extremely important. The second thing is that there are several structures and the circuitries involved in this comorbidity. One hand, these studies are very important to better understanding the underlying mechanism, but it's also very important to develop non-pharmacological treatment, as we all know that in pain field, there are more and more focus on non-pharmacological approaches like the TMS, RTMS, TDCS. So I think these studies are very important to feed these type of research. And I would like to also emphasize that it's very important that we focus on the sexual dimorphism in all our research. This is what we observed in our neuropathic pain model, that even though the behavior seems to same, the dynamic is completely different. And actually in my lab, we are focusing on this part and try to better understand underlying molecular mechanisms. And also what I would like to say is that today, because of the time, I just present you what is coming from the chronic pain to depression. I mean, that not really the vicious cycle, but we are also interested in the impact of the chronic stress on the pain chronification. And we published with Michelle Roche and David Finn from Galway recently a paper on that, and luckily we got a grant, and we will continue to work on that, and we really want to work on this visual cycle, meaning from chronic pain to mood disorders, but from mood disorders to chronic pain. So I would like to thank all my previous students worked on these projects, my collaborators, and the financial supports, and you for your attention. And if I may, I'm looking for a postdoc if you're interested. Good research, wine and cheese. You're all welcome.
Amazing plug. Thank you, Dr. Yalcin. Great work. So there is a question in the Q&A. I think we'll save it for the end just so we can make sure we have enough time. And then Dr. Finn, if you want to start sharing your slides as I introduce you, and then we'll get to the additional questions at the end. So up next, we have Dr. David Finn, who is an established professor of pharmacology and therapeutics and founding co-director for the Centre for Pain Research at University of Galway, Ireland. His research focuses on the affective and cognitive dimensions of pain, stress-pain interactions, and neuroinflammatory processes, with an emphasis on the endocannabinoid system. He's past president of the International Cannabinoid Research Society and of the Irish Pain Society, and was a member of the task forces for cannabis, cannabinoids, and chronic pain for both IASP and the European Pain Federation. Professor Finn is a member of the editorial boards of multiple international scientific journals, including Pain. He's published over 200 peer-reviewed journal papers and book chapters, and frequently lectures at international conferences. Thank you so much for being here, Dr. Finn.
Oh, thank you very much, Laura, and to the entire PRF Webinar Series team for this very kind invitation, and great to be here. So I'm going to talk about the role of the endocannabinoid system in pain, negative affect interactions. Going to focus mostly on preclinical data from our group and others, but finishing with some clinical evidence from our research towards the end. And you've already had a great introduction to this whole topic from Andrea and Ipek, so I make my job easy here. But as you've heard, there are reciprocal, bidirectional relationships between pain and negative affect, and we've heard about the comorbidity between chronic pain and anxiety and depressive disorders. We know that there are also common pharmacological treatments used to manage both chronic pain and affective disorders, like gabapentinoids, tricyclic antidepressants, for example, and of course that there are shared neural substrates that are commonly implicated in both chronic pain and negative affect. And in my lab, we are interested in both directions. But I'm going to focus in this talk mostly on the direction shown here, so how stress and negative affect impact on pain. So Ipek, you set this up nicely for me, thank you. And when we look at that, really the impact of stress and negative affect on pain depends on the nature, duration, and intensity of the stressor or negative effect modality. So stressors that are acute, intense, immediate threats to homeostasis tend to suppress pain through the phenomenon of stress-induced analgesia, whereas stressors that are more moderate in nature, that are repeated, that are future-orientated, anxiety, depression, repeated stress, tends to do the opposite and exacerbate pain through the phenomenon of stress-induced hyperalgesia. So again, in my research group, we're interested in both of these phenomena, and particularly interested in the role of the endocannabinoid system therein. So what's the endocannabinoid system? I'm sure it's familiar to most of you, but just as a quick recap, It's a lipid signaling system, very important signaling system, physiological signaling system in the body. It's comprised of at least 2 receptors, the CB1 and CB2 receptors. These are GI-GO protein-coupled receptors. And then we have the endogenous ligands for the cannabinoid receptors. The 2 best characterized endocannabinoids are anandamide and 2-AG. And then there's a whole enzymatic machinery that either synthesizes or degrades these endocannabinoids. For example, FA, fatty acid amide hydrolase, the primary degradatory enzyme for anandamide and related N-acetylethanolamines. Monoacylglycerol lipase, or MAG lipase, is the primary degradatory enzyme for 2-AG. And then there are also a whole host of non-CB1, non-CB2 receptors that are also targets for cannabinoids and endocannabinoids. And it happens that we find all of these components of the endocannabinoid system distributed throughout pain and affective circuitries pathways. So the receptors, the cannabinoid receptors, the endocannabinoids themselves, the enzymes are all to be found in primary afferent neurons, in the DRG and dorsal horn of the spinal cord, and throughout the brain, in these brain regions that are so important, as you've just heard, in pain and negative affect. And particularly, we find the endocannabinoid system has a strong distribution and expression throughout the descending pain modulatory system. So we're interested in the role of the endocannabinoid system in both stress-induced analgesia and stress-induced hyperalgesia. I'm going to start off by presenting some data on stress-induced analgesia and the role of the endocannabinoid system there. And why is this important? Well, stress-induced analgesia is a form of pain suppression expressed during or following exposure to stress. It's a really potent form of endogenous analgesia, actually. In rodents, when we model it, we see up to 90% suppression of pain at its peak. And it's also expressed in humans as well, so it has some translational validity and protective value. It is strongly mediated by the descending inhibitory pain pathway. We've known this for many years now, and we reviewed the literature in this review some years back. And in initial work that I did on this, actually when I was a postdoc way back in the early noughties, working with Vicky Chapman at the University of Nottingham, we were able to show that in a model of conditioned fear-induced analgesia, so conditioned stress-induced analgesia, that if we block the CB1 receptor with the antagonist rimonabant, it completely prevents the expression of fear-induced analgesia. Then in Galway, continued on with this work, and we showed in Galway that if we do the opposite, if we give a drug that boosts the levels of endocannabinoids, so this is a FA inhibitor, URB597, so inhibits the enzyme that breaks down anandamide and related N-acetylethanolamines. If we give that drug, it enhances fear-induced analgesia, almost to the point where formalin-evoked nociceptive behavior is completely abolished in these animals. So during fear, conditioned fear, endocannabinoids are being released, they're mobilized, they bind to CB1 receptors and mediate the antinociception in the formalin test. And we went on to look at the neural substrates involved here. We were able to show that fear-induced analgesia was associated with elevations in anandamide in the dorsolateral PAG and in the basolateral amygdala, and that if we pharmacologically block CB1 receptors directly in these brain regions, we can attenuate fear-induced analgesia. So fear-induced analgesia is a form of conditioned stress-induced analgesia mediated by the endocannabinoid system in the dorsolateral PAG, basolateral amygdala. We carried on and did other work, and if you're interested, these are some of the papers, but we were able to show that there's also a role for the ventral hippocampus and prefrontal cortex in this phenomenon also. And this, I think, work nicely complements the work of Andrea Homan and Daniela Piamelli, Jeff Mogul, and others who've shown a role for the endocannabinoid system in forms of unconditioned stress-induced analgesia as opposed to conditioned fear-induced analgesia, which is what we were more focused on in that work. Now, we're all, of course, aware of the importance of including sex as a biological variable in our preclinical research. We highlighted this in our paper from the PainDiff Consortium published earlier this year in Nature Neuroscience. This international consortium was led out of Galway here by Michelle Roach, together with myself and Brian Maguire. And this is important, and it's important in the context of what I'm talking about here, because there are sex differences within the endocannabinoid system of relevance to pain. There are sex differences in the expression of components of the endocannabinoid system throughout pain circuitry and negative affect circuits. And also there are differential responses of the sexes to cannabinoids and endocannabinoid system modulators in pain models. So the earlier work I presented to you as by way of background was in male animals, but moving to both sexes now, and here we can see that in Sprague-Dawley animals, fear-induced analgesia is more robustly expressed in males than it is in females. And there's a reduced expression in the female rats. And this is associated with differential levels of oleoethanolamide, a lipid related to the endocannabinoid system, in the PAG. So males expressing fear-induced analgesia exhibit an increase in the levels of OEA in the PAG, but we do not see this in the female rats. So that work that I've been talking about on fear-induced analgesia implied a model of conditioned fear and looked at in the formalin test, which is a relatively acute or short-term tonic inflammatory pain model. But we were interested in moving on and asking, well, what would be the effect of acute psychological stress on nociceptive behavior in a rat model of chronic pain, and specifically chronic neuropathic pain? And would we see sex differences here, and is there a role for the endocannabinoid system? So this is work that Stephanie Burke in my group did during her PhD, and very briefly on the methods, we're using the spared nerve injury model of neuropathic pain here, male and female groups. And at day 21 post nerve injury, the animals are exposed to 30 minutes of restraint stress, or controls are left in their home cage. And then an hour after the restraint stress, we do testing for somatosensory sensitivity, mechanical hypersensitivity, cold hypersensitivity. I'm just going to focus on the mechanical hypersensitivity data today for reasons of time. And so what we see, well, first of all, as you'd expect, the spared nerve injury does result in mechanical hypersensitivity on the ipsilateral hindpaw. No surprises there. And when we look at the effects of restraint stress, it doesn't have any effect in the sham animals on paw withdrawal thresholds in the von Frey test. But in the SNI animals, it does, restraint stress attenuates the SNI-induced reductions in paw withdrawal thresholds. So it basically attenuates the SNI-induced mechanical hypersensitivity in rats. So evidence of stress-induced antinociception. And actually, it's more pronounced in male animals than it is in female animals. Post-mortem in that experiment, we measured endocannabinoid levels. We measured them in lots of different brain regions. The region that sort of gives the most robust effects was actually the hypothalamus, which is important also in descending modulatory pain control. And we found a stress by surgery interaction effect for anandamide in the hypothalamus. And then looking more closely, and when we pool the sexes here, we find that there is an increase in levels of anandamide in the hypothalamus associated with this stress-induced antinociception in the SNI animals, which we don't see in sham controls. So this gave us a clue that maybe the endocannabinoid system is important in this model also of stress-induced antinociception. And so we carried on with a second experiment, which had a very, very similar design to the first one, except here now we are giving a single acute injection of CB1 receptor antagonist, inverse agonist, AM251, just after the restraint stress. So we're asking, what's the effect of pharmacological blockade of CB1 receptors on this form of stress-induced antinociception? And the answer is that it completely attenuates the stress-induced antinociception that we see with this restraint stress coupled with the spared nerve injury model. So it appears to be a CB1-mediated form of stress-induced antinociception also. So just to sum up for this first part of the talk, fear-conditioned analgesia, which is an important endogenous form of analgesia, is mediated by the endocannabinoid system and throughout multiple brain regions, particularly the descending modulatory pain system. And acute restraint stress also induces antinociception in the spared nerve injury model of neuropathic pain. That's behaviorally is more pronounced in males than in females, and appears to be associated with increased levels of anandamide in the hypothalamus and can be blocked by CB1 receptor antagonist. Okay, so moving from stress-induced analgesia to stress-induced hyperalgesia now. And is there a role for the endocannabinoid system here? For this work, we used a model that we've published on quite recently, actually in collaboration with Ipek. It's this paper here in Neuropsychopharmacology, just published a few months ago this year. And it is a model which looks at the effects of repeated restraint stress for 21 days, 6 hours a day on hindpaw incision-induced post-surgical hypersensitivity. And in this particular paper here, we showed a role for beta adrenergic receptor signaling and NLRP3, IL-1 beta signaling. I'm going to not talk about that and focus more on whether there might be a role for the endocannabinoid system in this model. And here are some of the results, first of all, in male Sprague-Dawley rats. So again, it's chronic repeated restraint stress, 21 days, 6 hours a day. And then after that, we carry out the hindpaw incision surgery, model of post-surgical pain, and we look at pain-related behavior after that. And this is a restraint stress, chronic restraint stress model, which results in reduced weight gain and depressive-like behavior in these male animals. And for pain behavior, you can see that it results in an exacerbation and a prolongation of both mechanical hypersensitivity and heat hypersensitivity. In a subsequent experiments, we looked at the effects of acute systemic administration of a FA inhibitor, the URB597 again, in this model. And it attenuated this exacerbation of the surgery-induced mechanical hypersensitivity by repeated restraint stress, suggesting a role for FA and FA substrates as potential therapeutic target here. And the same for the monoacylglycerol lipase inhibitor, MGN110, MAG lipase inhibitor. This is the enzyme that degrades 2-AG. So basically, elevating levels of 2-AG, it also attenuates this chronic repeated restraint stress exacerbation of surgery-induced mechanical hypersensitivity in male animals. We have also been doing experiments in female Sprague-Dawley rats at an earlier stage with this, but again, we see the same sort of behavioral phenotype where there's a repeated restraint stress-induced exacerbation or prolongation of the mechanical and heat hypersensitivity in female animals. And post-mortem, interestingly, what we see is a stress by incision, hindpaw incision interaction effect for anandamide in the prefrontal cortex, whereby restraint stress increases anandamide levels in the prefrontal cortex of sham animals, but not incision animals. And so we're interested to follow up on that finding and investigate whether it has any causal implications behaviorally. Just by way of, I suppose, a reminder, we've also been working over the years on the Worcester-Kyoto rat model. This is a genotype that exhibits anxiodepressive behavior, and it also exhibits pain hyperresponsivity. And some years back now, we showed that this hyperalgesia associated with an anxiodepressive phenotype in the Worcester-Kyoto rat model is associated with a deficit in endocannabinoid signaling, particularly in the RVM brainstem. So repeated restraint stress before hindpaw incision exacerbates and prolongs surgery-induced somatosensory hypersensitivity in both sexes. We can attenuate that exacerbation by FA and MAG lipase inhibition in males, and still need to do that experiment in females. And we see sex-specific alterations in levels of anandamide in the prefrontal cortex, seeing that in females but not males. So for the last part of my talk, just with one eye on translational relevance, asking the question whether there is a role for the endocannabinoid system in pain negative affect interactions in humans with chronic pain. And we had shown some years back that, of course, there are higher rates of depression and anxiety, higher scores in patients with neuropathic pain versus age-matched controls and sex-matched controls. This has been shown by many research groups over the years. We know this. We'd also shown in a cohort with burning mouth syndrome that there are alterations in plasma levels of N-acetylethanolamines and cannabinoids, and that these are correlated with depressive-like behavior in this burning mouth syndrome condition. But more recently, in collaboration with Patrick McHugh at the University of Huddersfield, Stephanie Burke in my group generated data and published this paper where we again show that there's higher anxiety and depression scores in patients with neuropathic pain versus controls. So that's okay, nothing new there. But interestingly, we did find that there are higher circulating levels of plasma 2-AG and lower expression of the gene that encodes for MAG lipase and NAPLD in patients with neuropathic pain versus healthy controls. So alterations in the endocannabinoid system. And that when we carry out logistic and multiple linear regressions, we see that the likelihood and severity of neuropathic pain can be predicted by the combination of age, sex, depression, anxiety, and one's endocannabinoid system status. So just to sum up there, that we see higher levels of depression and anxiety in patients with neuropathic pain, and that's accompanied by alterations in the endocannabinoid system and circulating levels, and that possibly blood endocannabinoid system status coupled with other clinical parameters, including mood-related parameters, may predict the likelihood and severity of neuropathic pain. So just before we finish, what's the therapeutic potential here for the endocannabinoid system? Well, there have been some RCTs on the effects of cannabinoid drugs on in people with comorbid pain and mood disorder. There aren't that many studies. For the most part, the ones that are published do show reductions, cannabinoid-induced reductions in both pain and the negative mood. But these are small studies for the most part, primarily with THC or synthetic derivatives or forms of THC or combinations of CBD and THC. So whilst it looks promising, we definitely need higher quality evidence and evidence with other types of endocannabinoid system modulators as well, like FAH inhibitors, MAG lipase inhibitors, other interesting ways to modulate the endocannabinoid system. The other interesting line of evidence that's relevant here, I think, is the evidence for polymorphisms in genes that encode for components of the endocannabinoid system, particularly the FAH C385 ALL, where this has been associated, SNPs in this gene have been associated with pain and negative affect. And I think is interesting further evidence for importance of FAH as a potential target for comorbid pain and mood disorders. So just to finish up, stress and negative affect have profound effects on pain sensitivity, but these are dependent on a number of different factors, including the nature, duration, and intensity of the stressor, one's pain status, and sham versus chronic pain. We see differential effects. And of course, sex dimorphism is also playing an important role. But within the descending pain circuitry, endocannabinoid signaling mediates antinociception induced by psychological stress in both inflammatory pain and neuropathic pain. Still have more work to do to elucidate the neural substrates there for neuropathic pain. And we see that in contrast, repeated chronic restraint stress exacerbates pain-related behavior, and this can be attenuated by FAH and MAG lipase inhibitors. In the clinical evidence I showed you, we saw that blood endocannabinoid system status combined with clinical parameters may be of some use in predicting the likelihood and severity of neuropathic pain, and that hopefully the data can inform the development of endocannabinoid system-based biomarkers or diagnostics and therapeutics for chronic pain and its comorbidity with affective disorders. So I'll just finish by thanking lots of people in my group who've carried out this work. Also collaborators, particularly Michelle Roach here in Galway, who I work closely with, and Ipek. Some of this work has been in collaboration with Ipek, Rohini Cooner at Heidelberg, Patrick McHugh in Huddersfield, and Eric Downer at Trinity College Dublin, and thanks to the funding agencies. We also are looking for postdocs, so I'll put that plug out as well. We have a new grant with Ipek and other colleagues around Europe, ERANET grant in this space of stress pain interactions, ERANET Neuron. And so if you're interested, please get in touch.
Thank you so much, Dave. Really interesting work. I know we're a bit over time, so I appreciate everyone hanging on. I do want to just pose the last two questions and try to keep it brief. So for Dr. Yalcin, did you find any network level effects of nerve injury in your fMRI data? This is from a graduate student also doing preclinical fMRI.
Perfect. Yeah, we do. I mean, it's very interesting that because as we did this, we scanned the animals before the surgery, 2 weeks after the surgery, and 8 weeks after the surgery. And we can clearly see that in the very early stage of the neuropathic pain, we have the network that are involved in the sensory motor response. And then with the time, we can observe that default network altered, and then salience, and then more limbic and emotional pathway. So it is really time dependent, these network and level changes occur, but we do see very nice clusters of different brain rewiring at different time points of the development of neuropathic pain.
Thank you. I think this is for Dr. Finn, since there seems to be so much sexual dimorphism, related to glial cell activity. Do you think the endocannabinoids act via central glia cell in males and via distinct mechanisms in females?
Oh, that's a really great question. Yeah. I suspect there could be something in that, but we haven't yet looked in that in detail. We know that cannabinoid receptors are expressed on glial cells, on microglia in particular. They can regulate the release of cytokines from glial cells. So I think that's really worthy of investigation. We also showed a role for LLRP3 signaling in the model of repeated restraint stress-induced exacerbation of post-surgical pain that we published earlier this year that I mentioned. So so we know that cytokine signaling, pro-inflammatory cytokines released from glia, are playing important roles in stress-pain interactions. We know that cannabinoid receptors modulate those glial cells. And that there are sex differences in both glial functionality in pain and the endocannabinoid system. So, yes, it's possible, but I think further experimentation is required.
Okay, and last one here. While Dr. Finn's data could fit with both Dr. Nackley's and Dr. Yalcin's models as a modulatory mechanism, I'm wondering how Dr. Yalcin and Nackley view each other's models, which seem mechanistically distinct.
Well, I think we're definitely modeling different conditions. I mean, ours is a model of chronic primary pain, and Dr. Yalcin's is a model of neuropathic pain, but not to say that these mechanisms and are siloed to one model or the other. But I would like to just highlight the interplay between like the ACC and adipose tissue even. And I was just getting ready to email Ipek about that, because we know that the ACC regulates adipose dynamics, and I'm sure it's a bidirectional relationship, which would be something that would be interesting to explore. But I mean, I think one thing that kind of cuts across the models though may be inflammation, and we see inflammation is a key component of all different pain conditions, and inflammation and neuroinflammation, and I think that is can be critical to both pain and mood disorders. So.
Yeah, I agree with Andrea, because I would like to talk with you later also, because we do see the alterations in the anterior cingulate cortex, that genes that are related with the adipocytes. So we have the data, and I think somehow it seems some mechanisms are distinct. Also, we should pay attention that the the time that we are making the analysis and the time that you're making the analysis, because some of the data that we generated at the late stage, we do not find it in the early stage either. Because in the neuropathic pain, as you know, the inflammatory mechanism much more involved in the early stage, and the late stage, we do not find that strong neuroinflammatory response, at least in my model. This is the reason why I think that we really need to compare the same time point or same dynamic in different models. So this might also explain why we do not, it seems that we do not see the same thing, but because we do not check the same thing, most probably. But for the adipocytes, yes, we do see the alterations. Yeah, in the ACC, we do see the alterations. So.
All right, great responses. Thank you again, everyone, and for hanging on and for your time. We really appreciate it. And this will conclude this installment of PRF's webinar series. Thank you so much for being here, everyone.